This video is going to walk us through the steps of translation. Remember, translation is the process of making proteins. So another way of saying that is protein synthesis. So during step one, which is called initiation, we have to get all of the key players to the start site. In this case, the start site is going to be the start codon. Remember, codons are found on the messenger RNA, and the start codon is always AUG. So that means we need to get the messenger RNA to the ribosome, but the ribosome comes in two pieces, the large subunit and the small subunit. So during initiation, we're also going to have to get those two parts put together. Then we'll also need the tRNA because those are what brings the amino acids to the ribosome. So, as a quick refresher, the messenger RNA, think of that M as messenger, as the actual message telling the ribosome what the cell needs to build that protein. The messenger RNA contains codons, and each codon codes for a specific amino acid that the tRNA is going to bring. So how I remember that codons are found on mRNA is codon sounds a lot like code. The M in messenger RNA is like message. So you can think of the messenger RNA as a coded message. Okay, coded message. So the codons are on the messenger RNA. And that's going to be important because remember, the tRNA that's going to bring the correct amino acid to the ribosome, it contains the anti-codon, all right? And that's how it knows where to deliver its specific amino acid. And I'm going to illustrate this process in a separate video, the next video in this series. But just kind of remind you, codons are on messenger RNA, and anticodons are on tRNA. All right, so let's talk about how we get our key players together. So the first thing that's going to happen is the messenger RNA is going to bind to the small subunit of the ribosome. That is going to recruit the initiator tRNA that's carrying methionine. Because remember, the start codon is AUG, and that always codes for the amino acid methionine. So you should always have methionine as the first amino acid in your polypeptide chain. Okay, so we've got the messenger RNA first binding to the small subunit of the ribosome. Then the first tRNA carrying methionine is going to come and bind to the messenger RNA. If you look at this picture, you'll see how that's happening. The tRNA has the anticodon UAC, which is binding to the complementary start codon found on the messenger RNA, which is AUG. Once that has happened, now the large ribosomal subunit is able to come and attach to the small subunit. If you look at the picture, you can see two of these little binding sites on the large ribosomal subunit. You can see the A site and the P site. Now there's actually one more that you'll see later. But what I just want you to notice is that because the tRNA is already there, it's going to kind of help the large subunit align properly with the small subunit. So the large subunit is attaching in such a way that the initiator tRNA can bind to that P site, okay? So at this point, we have all three key players together. So now we need to start building the polypeptide chain. And so there's a lot of notes on here, but I want you to look at the pictures while I talk, because I think that's going to help make more sense than you trying to figure out what bullet point I'm on and things like that, okay? So as you can see in this picture, there's the start codon. I'm looking at the top picture. You see AUG, okay? And the transfer RNA has attached to it and brought methionine. We saw that in the initiation step. So now in this example, the second codon is CUA. So automatically you know what anticodon the tRNA must have, right? Because it must be complementary to the codon. So if the codon is CUA, what is opposite of that? GAU, right? Think of base pairing. So the tRNA that has the anticodon GAU, which should be carrying the amino acid leucine, knows that it needs to come in and bind second, okay? So that's what's happening. That's why you see the second tRNA. It's come in and binded to that A site on the large ribosomal subunit. So now we have our first two amino acids present at the ribosome, but they need to be connected, right? And so that's essentially what happens. Notice that the methionine, excuse me, that was originally on our first tRNA, it gets scooched over to the second amino acid. In this case, it's leucine, okay? So now look at the bottom picture. Do you see how that second tRNA now has both amino acids attached to it? So what this means is we now have that first tRNA that's just empty, right? There's no more amino acids attached to it. That tRNA has done its job. So it's actually going to get released from the ribosome so it can go pick up another methionine for whenever another methionine is needed, okay? Now, when you are looking at these PowerPoints, when you see AA, sometimes I put A period, sometimes I just put AA. That means amino acid. It's just shorthand, okay? All right, so now we have this second transfer RNA with two amino acids attached to it. And what's going to happen is this tRNA needs to get out of the way so that the tRNA carrying the third amino acid can now come into the ribosome. So if we look at the picture, and you're looking at the bottom one, okay, because that's the picture on the top now is what we just saw on the bottom of the previous slide, okay? It's just there again to kind of remind you where we ended on the last slide. So now, again, looking at the bottom picture, we have that second tRNA that's now carrying both of our amino acids, amino acid 1 and 2. Now we need amino acid number 3 to come in. So here comes that tRNA carrying the next amino acid. But let's review how that tRNA knew that it was its turn, that it needed to report to the ribosome. So we have to look at the third codon on the messenger RNA, GCC. And so if you looked at the codon wheel, GCC codes for alanine. So the tRNA carrying alanine is the one that needs to report to the ribosome. And it's able to do so because of its anticodon, right? So if the codon on the messenger RNA is GCC, then the anticodon on the tRNA is going to be opposite that. So it will be CGG, which it is, okay? That's what you see on the tRNA here. So it's going to bind to this empty A site on the ribosome, and it's carrying that alanine, okay? So then what's going to happen, just like we saw on the previous slide, the transfer RNA that currently has methionine and leucine on it, it's going to transfer those two amino acids over to the new incoming tRNA, so that will now have methionine, leucine, and alanine, okay? So then that second tRNA will then be empty. It'll be kicked out of the ribosome to go pick up a new amino acid, and this process will continue, okay? And that's going to happen until all of the amino acids have been added. And again, there's usually at least 500 of them. Sometimes, 1,000 or so. So this is going to keep going. It's going to keep reading the messenger RNA keep having transfer RNAs come in and deliver those amino acids until a stop codon is reached, which is what you see on the far right of this picture. Now remember, there is no amino acid called stop. That means no transfer RNA is going to recognize that codon. Because remember, transfer RNAs always come attached to an amino acid. So if there's no amino acid called stop, there's no transfer RNA that's going to recognize that codon. So what happens? Okay, and here's something you're going to want to write down because I don't see the word on this slide. So please write this down. So what's going to bind in that free space on the ribosome where there's a stop codon? Something called a release factor is going to bind. And it's called a release factor because as you can see kind of now in the top right of the photo, the ribosome is going to separate or release from each other. That's going to release the messenger RNA. It's going to release the tRNA and the growing polypeptide chain. And that means this process is done. All the key players have released from each other. And now they're free to go and start translation on another piece of messenger RNA. Okay? So again, when there's finally a stop codon on the messenger RNA, a release factor binds to the ribosome, and excuse me, that's going to cause everything to release from one another, thus ending the process of translation. It's also officially the end of the third step, termination. Okay? Now, in cells, in some types of cells, it's really important that the process of translation happens quickly because there are thousands of different there are thousands of different proteins in the cell, especially when we're talking about like eukaryotic cells. But there are tons of different proteins, and we need many copies of each of those proteins. So this process has evolved to essentially speed up and happen really, really quickly. And one way that's done is to have multiple RNA polymerases on a single DNA gene. So if you're looking at this picture, okay, you can tell on the left, it kind of blows up the whole process. The blue the blue line is the DNA, and the little orangish oval is RNA polymerase, which means it's going to be making RNA. And then you can see the little ribosomes attached to the messenger RNA. And essentially what's happening is there are 4 different RNA polymerases on one gene, one segment of DNA. So that's allowing multiple messenger RNAs for the same gene to be made. And then notice that there are tons of different ribosomes on each messenger RNA. So in this picture, I don't know if I can see all of it because of the transcript some of it, but on that top RNA polymerase, right, it went first, so it has more of the messenger RNA made. It's got 1, 2, 3, 4, 5, 6, 7, 8 ribosomes, all creating the same protein off of that messenger RNA. The second RNA polymerase has, I think, what is that, 7 ribosomes attached. So basically, by having multiple RNA polymerases going at the same time and multiple ribosomes on each of those messenger RNAs at the same time, that's just how to make lots of protein very, very quickly. Okay? All right, that is the end of this video. I don't know if you could hear Kenny making all of his noises. He is letting me know it's time for breakfast. So y'all, please let me know if you have any questions.